Our next speaker is Watanabe, who is going to tell us about measuring exercise in Drosophila using a rotating exercise quantification system.
All right, so thank you very much for the introduction and for allowing me the opportunity to present my research to you guys today. So what I want to talk to you guys about is the rotational exercise quantification system. It is an exercise machine for Drosophila, and it's able to specifically quantify exercise in real time. So before I get into the nitty-gritty and the details, I just want to quickly remind you guys of why exercise is such an important topic in research today. So as we're all aware, there are many, many benefits to exercise, right? It is the most prescribed treatment method, along with a change in diet for weight control and obesity, which have been growing problems in the United States for the past several decades. It can strengthen bone and muscle, as well as reducing the risk or alleviating the symptoms of a whole host of diseases, including cardiovascular disease, metabolic syndrome, type 2 diabetes, and that list kind of just keeps on going on and on. I think it's also interesting to note that roughly 50% of Americans are meeting the CDC's guidelines for aerobic exercise, which was surprising to me because I don't. It's a very popular activity as well. Now, I think we all know that everyone is kind of aware that the effects of exercise depend on who you are, right? Or specifically, what genes you have. Now, despite the fact that we understand that, we actually have a really limited understanding of the whole overall genetic architecture that's underlying exercise response. There are some limited pathways known in humans. A particularly prominent one is PGC1 alpha, which I think some of you have heard of. It's part of the PPAR pathway, and it's been implicated in mitochondrial biogenesis, musculoskeletal structure, and a host of other downstream pathways. But again, this is all very limited compared to what we believe is everything that affects exercise response in the genetic architecture. On top of that, there's actually this new field of research that's coming in, which is known as exercise resistance. So these are individuals who are unable to improve their metabolic profiles based on exercise. We actually know almost nothing about that. So it's an extremely exciting time for us to be in exercise genetics research right now. So I'd like to switch gears here and move into a little bit of why we should be studying this in Drosophila. So in order to go into that, I'd briefly like to talk about some of the challenges of exercise genetics research in humans. So if I could direct your attention over to my figure here. So the physiological response, an individual's physiological response to exercise is going to be based on a couple of factors, largely the genome and the environment. So some factors that are going to affect the genome include pleiotropy, epistatic interactions, as well as epigenetics. Some factors that are included in the environment, so that's going to be everything. But some pretty big factors for exercise response include drugs, diet, circadian rhythm, motivation, and that list kind of keeps on going on and on. So the thing in human studies is that it's really difficult, right, to standardize the genome because we're all different for the most part. And the environment is also extremely difficult to standardize. Now, as you guys know, in Drosophila, for the most part, we can standardize the environments to a very decent level in the laboratory. There are a host of isogenic lines available, so genetic tools available to us, which means we can do multiple biological reps in our experiments. And there's also a wide availability of mutants, and we can obviously generate our own mutants. On top of that, there's also actually the metabolism is also extremely highly conserved between human and Drosophila. And on top of that, there are actually homologs for known human genes known human exercise genes that are also found in Drosophila and have conserved functions. So a little bit about how do we exercise flies. So to talk about these machines, I briefly want to remind everybody of a trait that Drosophila have called negative geotaxis. All that means is that flies are always going to want to go against gravity, so up. And they're always going to want to move that way. So the first Drosophila exercise machine developed was the Power Tower, which was developed by the Wessel's lab. And how this works is if you take a look at figure A, so fly vials are placed onto a platform, as shown here. And that platform is going to be raised up. And then, as shown in figure B, the platform gets dropped down. Now, when the platform drops down, all the flies in the vials are going to fall to the bottoms of the vials, and then they're going to crawl back up the walls due to the negative geotaxis. And that's just repeated over and over again, and so they keep on climbing up, dropping down, climbing up, and it sort of simulates an aerobic exercise. Now, the other apparatus that's been developed is known as the treadwheel, which was built by our collaborators at the University of Alabama at the Reed Lab. How this guy works is that, so the fly vials, or a vial filled with flies, is going to be put into the clamps that are shown here. And you can see how it's sort of attached to a rod. Now, that rod is going to rotate around, so the vials are going to be rotating 360 degrees, and the RPM is settable to whatever you want it to. So, and this induces exercise in that way. So if you'd like to learn a little bit more about the treadwheel, Dr. Reed has a poster here at the conference, and I've listed the poster number at the bottom right corner, so definitely go check that out. So the treadwheel and the power tower have really shown that exercise in flies can induce changes in metabolites, gene expression, feeding behavior, mass, and all of these studies, most of these, excuse me, are actually mirroring or very similar to trends that we see in human exercise. So they've really sort of established Drosophila as a solid model for studying exercise genetics. Now, the one component that I felt that I could add to the existing tool set here was a quantification method for exercise in Drosophila, because this was something that didn't exist yet. So this is something that's done in human and mammalian models as measures of fitness, for example. So when looking at humans, for example, oxygen consumption is measured against the amount of work done on a treadmill, and that is used as a measure. A measure of physical fitness is VO2 max, which is extremely commonly used, and that is measured when oxygen consumption reaches a peak and then remains steady, despite the fact that there's an increase in the amount of workload. So the thing that was that when I was looking at this, there was actually no comparable measurement for the exertion in the fly exercise. It was currently done by like amount of time run, for example. So I wanted to develop a system that would be able to quantify exercise levels in Drosophila. And so we came up with the rotating exercise quantification system, or the REQS for short. So how this guy works is there is the unit in the center right here. This is a locomotor activity monitor 25 high sensitivity model from Trikinetics. And in itself alone, it's used to measure activity levels in Drosophila and other small creatures. How it works is that there is a laser going through the middle of the vials, and every time there is a crossing, or a laser crossing, that's recorded as an activity point. So my basic thought process here was, if I can just stabilize these vials into the machine, into the LAM25H, and then rotate the entire machine around, right, then I should be able to induce exercise in an analogous way to the treadwheel, while simultaneously being able to record real-time activity levels. So kind of like this, and just a continual rotation going around. So we decided to do, so we built this rotational, we built this custom rotator for the REQS, for the LAM25H. And then we decided to do a couple of experiments to characterize this machine. So in our first experiment, we measured basal activity levels as well as induced activity levels, firstly to see if we're actually able to induce exercise in our Drosophila through our mechanism. And we did this in 4 genotypes, also to be able to see if we could record differences in activity levels based on genotype in Drosophila. So taking a look at our results here for these graphs, so the y-axis is just going to be mean activity level, and the x-axis is going to show the line as well as the sex of the flies. Each genotype is a different color on these graphs, and the females are going to be the first bar that shows up. So what we were able to find is that there was obviously a difference in activity by genotype, which I think you can tell just by looking at a few of these lines, both in the basal activity as well as in the induced activity. For example, looking at line 181 compared to line 73, for example. There were clear differences there. We also found that there were differences in activity by sex. Specifically, we found that males were exercising more than females in these 4 lines. However, this was also dependent on genotype. For example, looking at line 181, we can actually see that the females are exercising more than the males. But for the other lines, the males are exercising more than the females. We also ran ANCOVA, and we used basal activity as our covariate, and we wanted to see if induced activity was going to be greater than basal activity, and we found that to be true. So there was a significant difference there. However, this was also dependent on genotype again. So from this experiment, I think we can see that the REQS is really able to induce activity and at the same time record those activity levels in real time. For our second experiment, we wanted to see if we could kind of use the REQS to compare a couple of different exercise regimes to each other. So we could construct different exercise regimes in Drosophila. So we tried two different exercise regimes. We did a 2-hour continuous exercise regime where the flies were continually exercised for 2 hours. The rotation was just kept on. And then we did an interval training method. So this was done where the flies were exercised for 30 minutes at the beginning and then rested for 10 minutes, exercised for 20 minutes, and then rested for 10 more minutes, and then exercised for a final 10-minute period at the end. And we looked at this in 3 different genotypes than the ones that we used last time. So our results show, looking at the 2-hour exercise here, I guess really quick, so looking at this graph, each genotype is again a different color. This time the males, though, are filled in, or the filled-in shapes, and the females are going to be the open shapes on the graph. So what we found from the 2-hour exercise was that the flies mostly, so lines 852, or I'll just use colors, the green line and the red line, you can see that there was a slow reduction in activity level as the exercise went on, presumably because the flies were getting tired. Now, with the blue line, however, we can see that they sort of start to exercise as it goes on, and then they just kind of level out. I kind of assume that that's because they never really exercise very much because they're a lazy line, so they just kind of are like hanging out but doing a decent amount, but they're not really getting tired because they're not very moving around in the first place. Now, looking at our interval training method, we saw a couple of things. So the first thing that we saw was that if we give the flies a 10-minute rest period, so you can see that the rest period happens here, that immediately afterwards, the flies were actually able to recover almost their full activity level from as the previous time point. So on top of that, what the interval training really showed us was you can see that each of the lines kind of follows this curve-like structure, although they're different in magnitude. Sorry, I'm really bad at moving this mouse. And you can kind of see that the machine really does induce exercise, and that when the machine is turned off, that the flies are not exercising, that there's very little activity going on. So I hope from these 2 experiments that I've been able to convince you guys that the REQS is really able to quantify activity levels in Drosophila, and that we're able to record that in real time. So for future applications of the device, so obviously the main thing is that it's going to be used to quantify exercise in Drosophila. Now, some applications for that include the comparison of different exercise programs, diet and exercise combination experiments, behavioral and neurological studies that can also be done relevant to exercise. So with that, I would like to thank our funding sources, particularly the Nutrition Obesity Research Center, as well as the Center for Clinical and Translational Sciences at UAB, who helped us out with the statistics, all of the members of the Riddle Lab who have supported me, as well as the DeLuca Lab and the Reed Lab for their support. I would also like to briefly mention that our lab is currently looking for a postdoc. So if you're interested in studying epigenetics in Drosophila, particularly the heterochromatin, HP1, if you could contact either myself or Dr. Nicole Riddle at UAB, that would be fantastic. Thank you so much for your time, and take any questions.
We'll just take one quick question, so we're running slightly late.
So I noticed in your comparison of the 2-hour continuous versus interval training,
Yes.
that one of the lines
Yes.
suddenly responds where it didn't in the 2-hour, and yet that trial is the same at the beginning. So do you see a lot of variation from one trial to the next?
Yes, I believe there is a decent amount of variation in the trials, but it's not that big comparison to the whole, looking at it as a whole.
Oh, okay. I also want to point out that I am a poster child for the inherent risks of exercise at the moment.
Oh.